Yeah, sir, first, sir already joined the meeting.
Okay, okay. Thank you, Satya. Now we are moving on to the next season. I'm happy to share that our next honorable speaker, Professor Kunal Roy, has joined us, and the topic of his talk is in silico toxicology using q-RASAR and ARKA approaches. But before we begin, it's time for a grand welcome for him. So once again, I would like to invite the co-coordinator of this FDP program, one of our finest researcher and associate professor at Triple IT Allahabad, Dr. Sintu Kumar Samanta. Sintu sir, please give a warm welcome and a brief wonderful introduction of Professor Kunal Roy.
Oh, thank you, Sarpras. It's my great pleasure and honor to introduce our distinguished speaker, Dr. Kunal Roy, professor at the Theoretics and Cheminformatics Laboratory and former head of the Department of Pharmaceutical Technology, Jadavpur University, Kolkata. Dr. Roy is a highly respected name in the field of medicinal and pharmaceutical chemistry, especially known for his pioneering work in quantitative structure activity relationship (QSR) and molecular modeling. He has an outstanding academic background, being a gold medalist in both B.Pharm and M.Pharm from Jadavpur University, and later completing his PhD from the same institution. Over the year, Professor Roy has earned numerous prestigious fellowship and awards, including the Marie Curie International Incoming Fellowship at the University of Manchester, the Commonwealth Academic Staff Fellowship, and the AICTE Career Award and the DST Fast Track Young Scientist Award. His research contribution have also been recognized internationally with several most cited paper awards from Elsevier journals such as Bioorganic and Medicinal Chemistry and Bioorganic and Medicinal Chemistry Letters. He also served as the co-editor-in-chief of Molecular Diversity Springer and is a member of editorial advisory board of reputed journals including the European Journal of Medicinal Chemistry, Journal of Molecular Graphics and Modeling, Chemical Biology and Drug Designing, and Expert Opinion on Drug Discovery. Dr. Roy's research interests cover a wide range of topics from studying the QSR of pharmacologically active ligand and antioxidant to modeling physiochemical properties, ergochemicals, and environmental pollutants. His work has a remarkable impact on drug discovery and computational chemistry worldwide. We are truly privileged to have such a renowned scientist and academician with us. On behalf of everyone here, I extend a warm welcome to you, sir. We eagerly look forward to hearing your valuable insight and learning from your past experience. Over to you, Dr. Roy.
Thank you for nice introduction. Am I audible?
Yes, you are.
Yes, sir. Yes, sir.
Okay. And I think you can view my slides. I have already
Yes, sir.
shared
Yes, sir.
with you.
Yes, sir. Yes.
The topic is, as you already know, that is quantitative read across structure activity relationship, and ARKA approaches with reference to in silico toxicity prediction model development. Now, to start with, we are surrounded by a large number of chemicals. We are exposed to chemicals from our everyday uses of different kind of compounds, starting from the household uses of, say, biocides, pesticides, cosmetics, personal care products, pharmaceuticals, and other industrial products. The effect of hazardous chemicals and pollutants on the ecosystem is a matter of great concern because although we have a large number of chemicals in the environment to which we are exposed, relatively few of these chemicals have been subjected to adequate assessment for their hazardous environmental properties. Now, with the ever-increasing production of new chemicals and as the cost of experimental testing is rising, and also we have to consider the requirements to optimize resources for risk assessment of existing chemicals in use, the regulatory agencies from all over the world, for example, the US EPA, OECD, ARKA, European Chemicals Agency, etc., recommending alternative methods, including in silico approaches for ecotoxicological risk prediction. So as we have to give more emphasis on alternative methods to in vivo toxicity testing, as we have to reduce the animal experimentation. Actually, the animal experimentation is banned for some special kind of compounds. For example, cosmetics. For testing cosmetics ingredients, we cannot do animal testing. We have to rely on either in vitro or computational approaches. Under the REACH regulation, registration, evaluation, and authorization of chemicals, which is effective in European Union, more efforts are being given to include methods such as in vitro techniques and computational methods, including q-RASAR or quantitative structure activity relationship models. So these kind of alternative approaches are termed as new approach methodology. These are time and cost effective. These avoid animal experimentation. They support 3R principles: reduction, replacement, and reduction of animal experimentation. They can be applied to virtual compounds, and they are also supported by various regulatory organizations. Now, q-RASAR is an acronym of quantitative structure activity relationship, which deals with development of predictive models that correlate biological activity, which may be therapeutic or toxic, of chemicals, which include drug, toxicant, and environmental pollutants, with chemical structure information, which is mainly represented in the form of descriptors. So descriptors are nothing but numerical depiction of chemical structure information. And this kind of relationship is developed by the application of statistical tools. So the basic notion of any q-SAR is that biological activity is a function of chemical structure and or property. Or in other words, we can say that biological activity is the function of descriptors. Now, if we want to see some simple examples from drugs, we can give examples of some phenothiazine drugs like chlorpromazine, promethazine, and ethopropazine. Now, chlorpromazine is an antipsychotic drug, which has a side chain like this. Now, in case of promethazine, there is some change in the side chain. As you can see, there is a branching. And in case of ethopropazine, it's an anti-Parkinsonian drug. This is a diethyl substituent. So small changes in the substituent may change the therapeutic category. Again, tolbutamide and chlorpromide, they are structurally similar. They are sulfonylurea derivative. Now, tolbutamide is hypoglycemic, whereas it is short-acting, and chlorpromide is longer-acting. This occurs due to presence of the chloro substituent in chlorpromide. We can give other examples like isoproterenol and epinephrine. Isoproterenol is hypotensive, whereas epinephrine is hypertensive. And this occurs due to changes in the substituent on the nitrogen. In isoproterenol, it is isopropyl substituent instead of methyl as in epinephrine. So as you can see, with small changes in substituent, the type and magnitude of the biological activity may change. Not only changes in the substituent pattern, but also their orientation in space is also important. So stereochemistry is important. For example, diethylstilbestrol in trans form is more estrogenic. Similarly, propranolol is effective in the laboratory form. So here only the hydroxy group orientation in space is changing in propranolol structure, and that's why one form is active and another form is inactive. Similar case occurs in case of epinephrine, where minus laboratory form is more active. So stereochemistry is also important. So with changes in the substituent pattern, with changes in the structural features, with changes in stereochemistry, biological activity of a series of compound changes. And these changes occur in a very systematic way. And we can identify the pattern, and we can encode that pattern in the mathematical format. We can develop some models. The models can quantitatively predict the activity of compounds. And for developing such models, we should have a set of compounds with known the biological activity or some property values. And based on that, we can develop a model. And once the model is developed and properly validated, we can apply this model on new set of data for prediction parts. That means for compounds for which do not have experimental activity values, we can fill the data gaps by using model-derived predictions. And for data gap filling, we have different approaches like expert-based system or q-RASAR, read-across approach, which is basically a similarity-based approach. Based approach of structural arts. So today we'll now understand how we encode chemical structure forms. Chemical structure forms can be encoded in different ways. They can be encoded in the form of molecular graphs. They can be encoded in the form of molecular descriptors, which are numerical quantities to describe molecular structure information, or we can encode them in the form of fingerprints, which are mostly presented in binary form. We can also textual representation of chemical structure in the form of smiles or inch. Now, depending on the chemical structure representation, we can get molecular descriptors in different level, and molecular descriptors of different level have information of different dimensions. Now, from molecular formula and one-dimensional structure, that is the textual representation of chemical structures, we can get constitutional descriptors. From two-dimensional representation of molecular structure, we can get topological descriptors, structural, functional group counts, etc. From three-dimensional representation of molecular structure, we can get geometrical, electronic, and spatial descriptors. From surface structure representation, we can get molecular shape analysis descriptors. And sometimes the ligand protein interaction can also be used for computation of descriptors, but mostly for q-RASAR we use only ligand information, not the information from the target. So q-RASAR is essentially a ligand-based approach. So starting from appropriate molecular structure representation, we can get some numbers which can encode molecular structure information. So we can give some simple examples of molecular structure information, like say partition coefficient or log P value, molecular weight, molecular refractivity, pKa value, that is the acidic dissociation constant, and some simple structural information like presence of aromaticity or number of double bonds, number of carbon atoms, etc. So these are very basic level of structural information, but the structural information can be qualified at the higher level also, for example, water chemical descriptors. So we can calculate a large number of descriptors. So these descriptors are usually presented in the form of columns, and the compounds or data points are presented in the form of rows. So we will have several rows. Each row corresponds to a particular compound, and we have several columns. Each column corresponds to a particular descriptors. So we will have a large table, but we have to reduce the dimension of this table because there will be some redundant information. Some of the columns may be interrelated, or there will be some of the columns which may have no variance in their values. So you have to omit such columns. And also we have to consider that all columns or descriptors are not important for the endpoint or biological activity that you are going to model. So we have to identify the important features or descriptors for the particular biological activity. And this is done by several feature selection process. So after identifying the important features, we can go for model development. So models can be presented mathematically through equations, or the results can also be presented in a graphical way through contour maps. Some of the advanced machine learning models may not be presented through equations because the simple linear relationship is not present in those cases. Whatever may be the case, after development of the model, we have to judge the quality of the model developed through different statistical tests. And when we are satisfied that the model developed is of appropriate statistical quality, then we have also to see whether the model is able to predict the activity of new set of compounds. So that is done usually by the process of validation. So once we are satisfied from the process of statistical quality valuation and validation, then only the model can be applied to new set of data. And the model can also be interpreted properly for mechanistic explanation. And the developed model can also be used for designing new set of chemicals. So if we want to see the workflow of any q-RASAR modeling exercise, we have to start with the biological data collection. And biological data can be obtained from different sources like online databases or literature, some scientific articles, etc. But we have to take care about the quality of data that is being collected. Because if the quality of the data is poor, the developed model will also be of no use. Then the collected biological data and the set of compounds should be subjected to biological and chemical curation. Now, as the data are being collected from different sources, the same compound may be present from in different sources in different names. So we have to omit those duplicates. And in some of the cases, the same compound may be present in different databases with different biological activity values. In such case, we may choose to take the average of biological activity value of the same compounds, or if the values are differing too much, then we are less confident that which value is correct, that we may omit some or all of the compounds. And similarly, we have to check for some structural normalization, and we have to check for some curation like removal of inorganics, removal of mixtures, and usually if there is some kind of compounds which is present in very low quantity, some specific structural features are present in a low number or quantity, so those dissimilar structures may be omitted before starting the q-RASAR modeling exercise. So curation is very much important. At the beginning of q-RASAR, and after collecting the data points and curate calculation. As I mentioned, that descriptors are numerical structure prediction, so we can take help from different software for descriptor calculation, and there are different tools like AlphaDisk, Dragon, OChem, etc. for descriptor calculation. So after we calculate descriptors for the set of compounds that we have considered, we have to go for dataset division. So we have to divide them into training and test set. The training set will be used for model development purpose, whereas the test set will be kept aside. This will not be used for model development. This will be kept as unseen data set. So the feature selection and model development will depend on the training set only. The test set information will not be used. After development of the model, the developed model will be used for prediction of the test set data for which we have the experimental values, but we have blinded them. So the predicted values will be compared with the observed or experimental values. And when we see that the predicted values are close to the experimental values, that means residual is low, then only we can check the ability of the model to predict for the new set of data. So After we divide the data set into training and test, we have to go for feature selection using only training set data. And this we can do using a number of methods, including genetic algorithms, stepwise selection, etc. And after we have selected the important features, we have to go for selection of appropriate learning or algorithms. Learning algorithms may be of two types, supervised and unsupervised. In case of unsupervised, we do not use the endpoint data or the biological activity data. So unsupervised methods are used mainly for uh, uh, checking the data and pattern recognition. This can be done by clustering or dimensionality reduction. Whereas in case of supervised model development, we use the Y data, and there may be two kinds of model development. One is regression, other is classification. In case we have the quantitative biological activity values, we will go for regression model development. And in cases where we do not have quantitative values, but we have values like active, inactive, or high, low, we cannot use regression modeling strategy. We have to apply classification methods. So after we have developed our model, we will go for checking the quality of the model through different statistical tests, and then we will go for validation and an applicability domain estimation. Now here we have to mention that OECD has provided a set of guidelines for q-RASAR model development and prediction and validation. So we have to abide by these guidelines. The first guidelines says that there should be a defined endpoint that we are going to model. So all experimental values that are used for q-RASAR model development should have been obtained using same experimental protocol. There should be an unambiguous algorithm. So all the processes should be presented in such way that it can be reproduced with ease by other group of authors. There should be a defined domain of applicability. So our developed model may not be applicable for whole universe of the chemicals. They are applicable only for specific kind of chemicals which is similar to the training compounds. So if the query compounds are much similar to the query compounds, to the training compounds, in that case, that particular query compounds may not be predicted with reliability from the q-RASAR model. The fourth guideline says there should be appropriate measures of goodness of fit, robustness, and predictivity, and these are done using various statistical tests and validation criteria. And the fifth guideline is there should be a mechanistic interpretation if possible. Now, according to the OECD guideline, we may show the q-RASAR work in this way. As per OECD guideline 1, we have to collect the data from literature having a particular endpoint. And then we go for the next steps, that is structure, representation, descriptor calculation. And according to OECD principle 2, all the steps should be properly documented, reproducible. So data between the data set division, data processing, everything should be presented with clarity. Then according to OECD principle 4, there should be appropriate measures of the quality of fit and creativity. So we have to use different metrics to know the quality of the model, and which may be regression or classification based model. So we have different kind of metrics, different metrics for regression and classification. Be able to predict for new set of data. And according to OECD principle 3, we have also to check the applicability domain of the model to know whether the query compounds are within the domain or not. And then, according to OECD principle 5, there should be appropriate mechanistic interpretability of the model if possible. Now, in general, the regression, that is statistical models that are used for q-RASAR model development are of two types, regression and classification. In regression model, the simplest ones are method of least squares and partial least squares. And in case of classification models, linear discriminant analysis and logistic regression. Apart from this, there is increasing use of machine learning models, which make. Now the metrics for judging the quality of q-RASAR models depends on whether you are using the regression-based models or classification-based models. In case of regression-based models, we have different kind of metrics like, say, determination coefficient or R square, but the R square does not consider the number of features that are used for model development. So in such case, if we want to penalize a model for the number of features used, we have to use adjusted R square, where in the expression we can use the P, or the number of features that are used for model development. Then there are some error-based metrics like RMSE, which actually evaluate the degree of deviation between the observed and fitted values, and we have to take the square of the residual, and we take sum of the square of residual, divide by number of compounds, and we have to take square root. That gives RMSE. But these are these metrics that we have already mentioned, that is the R square, RS squared, and the RMSE. These are just quality of fit. They do not say anything about the quality of prediction. So to know the quality of prediction, we have to use different kind of validation metrics. So there are different kind of validation measures like internal and external. So I'll come to those. So any q-RASAR modeling technique should ultimately lead to statistically robust models capable of making accurate and reliable predictions. So the q-RASAR model development can be divided into 3 stages: data preparation, data analysis, and model validation. The validation strategies check the reliability of the developed models for their possible application on new set of data, and confidence of prediction can thus be judged. As I mentioned, the validation may be internal or external. In case of internal, we can do this by the method of leave one out, but we can also use leave many out. In case of cross-validation or internal validation, a segment of the training data is kept aside as internal test set, and from the rest of the compounds, the model is regenerated, and the left out samples are predicted from the regenerated model. And this is done in iteration. In the second iteration, some other set of compounds are kept aside, and from rest of the compounds, model is regenerated, and the left out compounds are predicted. In this way, we can find out the predicted values of different set of compounds. The simplest form, in each iteration we can omit only one compound, and this method is known as leave one out, but we can also calculate leave many out internal validation measures. Another method of internal validation is bootstrapping, where we omit some samples of compounds, and this can be done with replacement. So here subsamples are drawn in each iteration, and from rest of the compounds, model is generated, and the left out samples are predicted. And finally, you get the bootstrap R square value. In case of external validation, we do not use the training set data at all. We use the external set of compounds, and usually the external set of compounds, the additional compounds may not be available. So for addressing this issue, the original set of compounds is divided into parts: training and test set, as I have already mentioned. Test set compounds are kept aside, and after development of the model is used to predict the test set compounds. And then the predicted values are compared with the experimental values, and if the predicted values are close to the experimental values, then the model is good, and this is quantified in terms of R square trade. As you can see the expression here, the value should be close to 0.6 or higher. Now, in case of classification, we cannot use measures like q square LO or q square F1. We have to use different kind of metrics like sensitivity, specificity, accuracy, and precision. Those are derived from true positive, true negative, false positive, and false negative from contingency table. And we can derive F measure, Matthews correlation coefficient, and we can also judge their quality from the receiver operating curve or ROC. Now, if we see the application of q-RASAR, q-RASAR is applied in pharmaceutical science for lead optimization and drug development, ADME optimization. It can be used in material science for optimization of different properties. It can be used in food science, agricultural science, nanoscience, predictive toxicology, and for regulatory aspect. Now, another related data gap filling technique is read across, where q-RASAR is a statistical method. Read across is not a statistical method. It is based on hypothesis and is based on the similarity principles. So for the compounds without the experimental values, or for the query compounds, we have to identify its close congeners. And the definition of closeness may be based on the structural similarity, but we can also include other kind of similarity, like similarity in the metabolic profile or similarity in toxicity, etc. So based on defined similarity, we have reduced uncertainty. We have to give argumentation and justification of how we define the other compounds as close compound to a particular query compound, and which respect. And in this way, the confidence may be grown, and based on that, the hypothesis may be accepted for regulatory decision. So this is a purely non-statistical approach, but one can place this in a statistical modeling framework. So we have recently developed a framework of quantitative read across, where we put it in a quantitative framework. So starting from the data set, the data set is divided into two parts: source and query compounds. The source compounds are like training, and query compounds are like test. And the set of source compounds are again divided into sub-training and validation set. And the validation set is used for optimization of different similarity criteria, for hyperparameter of different kind of similarity in Euclidean distance-based, Gaussian kernel-based, and Laplacian kernel-based similarity definition. So once these hyperparameters are optimized from the validation set, we can use these optimized hyperparameters for prediction of query compounds to chemical read across through weighted average predictions. So once we get the predictions, then we can apply the usual validation metrics as are used in case of q-RASAR. Now, to give example, say we have a query compound like bromobenzene, and we want to know the endpoint value for bromobenzene from the similarity principle. And say, for example, we have identified some of the close congeners like say toluene, chlorobenzene, and phenol. So we have to see we have to check the similarity of bromobenzene with the close congeners like toluene, chlorobenzene, and phenol. And based on that, we will define kind of similarity, Euclidean distance-based, Gaussian kernel-based, and Laplacian kernel-based similarity. And based on this similarity definition, using these similarity values, we will calculate the endpoint value through weighted average prediction for bromobenzene. In this way, the read across prediction can be done for different compounds, and once we get the predictions, then the predicted values can be compared with the experimental ones, as we do in case of q-RASAR. Now, as I mentioned, the q-RASAR is a statistical approach, so it requires a good number of compounds for statistical reliability, because it needs a good degree of freedom for reliability. So when the number of compounds is very limited, we may not be able to apply q-RASAR to generate a statistically meaningful model. So in such case, read across may be useful, because read across does not require statistical model building. On the other hand, q-RASAR can give quantitative information for different features, which may not be possible in case of read across. So we can take the merits of both of the methods, q-RASAR and read across, and we can combine them in the same framework, and that is called read across structure activity relationship. So this is the workflow for our quantitative read across structure activity relationship. So here we have two set of compounds. One set is training, and other set is query, but we divide the training set into sub-training and validation set. And we define different kind of similarity measures, and we optimize the read across similarity measures through through prediction from the validation set. And once the optimized setting are obtained, we calculate read across structure activity relationship descriptors. So these are basically similarity and error based descriptors. I'll come to this. And once we calculate the RASAR descriptors, we'll combine them with the usual structural and physiochemical descriptors. And on the combined set of descriptors, we will apply feature selection. I will develop the model, and from the developed model, we will get the prediction from the test set compounds, as we do in case of q-RASAR. But RASAR is different from q-RASAR in some respect. I'll try to explain here. Now, say we have a set of compounds, and obviously different compounds will have different kind of biological activity values. So some compounds will have low values, some compounds will have higher values. So in the training set, we will have a mean of the training set response. So if we consider that mean as the threshold, so we can classify the training set compounds into two parts. So one set of compounds with values higher than the training set response mean may be considered as positive compounds, and the rest of the compounds having the response value lower than the training set response mean may be considered as negative set of compounds. So in this way, we can classify them. And for each query compound, we can identify their close compounds, and based on the activity values or response values of the close compound for a particular query compound, we can calculate different error methods like standard deviation, their coefficient variance, and standard error. On the other hand, we can we can calculate the similarity of closed source container for each query compound using different methods like Euclidean distance, Gaussian kernel, and Laplacian kernel, and we can calculate the similarity values. And from that, we can calculate average similarity, SD similarity, and CB similarity. Now, depending on the positive or negative compounds present in closed source container, we can also calculate maximum positive, maximum negative similarity values and their absolute difference, and positive average similarity and negative average similarity, etc. So these metrics are actually used, these measures are actually used as RASAR descriptors, and from that we can also get different derived descriptors that are shown at the bottom of this slide. I will not go into details. So this shows the basic RASAR descriptor list: SD activity, CB activity, average similarity, SD similarity, maximum positive, maximum negative, absolute difference. And we also use a concordance measure G based on the value of positive fraction, that is the fraction of the compounds present in the closed source containers for a particular query compound. Now, G or concordance measure has some ambiguity depending on its closed source containers and the positive fraction values, because for the same set of positive fraction value, it may show same G value for different sets of positive fraction values. So, for example, the G value will be 0.8 for when the positive fraction is 0.6 and also negative fraction 0.6. So we can address this ambiguity by defining another measure, that is GM. I'll come to that afterwards. Now we have also developed another composite function which is called RA function, which takes into account all contributing descriptors through the similarity definition, and we must combine the RASAR descriptors, that is similarity and error based descriptors, with the usual structure and physiochemical descriptors for getting more precision in the prediction. And the third the third picture here shows different scenarios where for a particular query compound which is shown in the form of a question mark, the closed source containers may be of different types, where most of the compounds may be positive. That means positive fraction may be 1 and GM value will be 1. Or the most of the closed containers may be negative. So maximum positive will be less than maximum negative and GM will be negative. Or they will be in the borderline where the closed containers may contain both positive and negative compounds. In this case, GM will be 0 So reliability of for such compounds will be low. So according to RASAR algorithm, we start with the chemical set and we get the feature vector in the form of similarity based on fingerprints or some other properties may also be possible. And we apply supervised learning technique for model development. For model prediction for multiple endpoints, and based on similarity, maximum similarity of the query compound to known inhibitors of different targets, we can link them to different molecular initiating events that are relevant for adverse outcome pathway. So RASAR algorithm has also a possibility to link to the adverse outcome pathway. But in the simple form, if we want to describe RASAR, which is a combination of q-SAR and data cross, starting from a data set, we compute 2D descriptors, but one can also compute 3D descriptors. And we divide the results into training and test. We select the features, we calculate, we develop the q-SAR model. At the same time, we calculate the similarity measures and combine them with q-SAR features. We develop the RASAR model by combination with similarity and q-SAR descriptors. I told that RASAR is different from q-SAR in several respects. In case of q-SAR, we calculate descriptors for a particular compound from the structure of that particular compound. Whereas in case of RASAR, we calculate RASAR descriptors not from that particular compound, but from its closed source congeners. Secondly, in case of q-SAR, we calculate descriptors first, and then we go for training and test set division. But in case of RASAR descriptors are calculated after division of the data set into training and test, because here test set compounds are used as query. So the RASAR descriptors are calculated by calculating similarity of the query compounds to different training or source compounds. So in this way, RASAR is different from q-SAR, and as we calculate RASAR descriptor based on the closed source congeners, so the prediction aspect will be inbuilt in RASAR workflow, and that's why in most of the cases we get that the external validation quality for RASAR models are better than q-SAR. Now we have performed several case studies. I'll give some examples here. The first one is from androgen receptor binding affinity. This is a collection of rat androgen binding affinities with 200 data points. So after structural curation, descriptor calculation, data pre-treatment, we go for data set division. Finally, we have a training set of 103 compounds and test set of 44 compounds. We select our features through genetic algorithm and followed by base subset selection. And finally, we develop our q-SAR model based on partial least squares. And in the right hand side, we have shown the model. As you can see, there is a mathematical equation. There is equal to sign. The left hand side, there is log RBA. This is the y value or dependent variable or the receptor binding affinity that we are modeling. But in the right hand side, you have several descriptors like S triple SCH, max A, CH, etc. So these are actually the information about the molecular structure. And we can see each descriptor is associated with a number with either positive coefficient or negative coefficient that describes the contribution of that particular descriptor towards the response. And followed by these, we see different kind of measures or metrics, like say the R-squared frame, as I already mentioned initially, the determination coefficient, then the q-score LO, that's the internal validation quality or false validation measure, and q-score F1, that is the external validation measure, the value of which should be more than 0.6. But here we are getting 0.58 in case of q-SAR model. So this slide also shows that the quality, that the source of the data is the KDB database. Then we have the 102 training compounds and 44 test compounds. And the same equation is shown here, where we find that q-score F1 value is 0.58, which is lower than 0.6. And these are the descriptors which are used during q-SAR model development that describes different structural features, but I will not go into details here. Now, when we apply chemical read across, which is a non-statistical approach, instead of q-SAR, we see that q square f1 is increased. That is, the external validation quality is increased. From 0.58 we get here 0.65. So using the optimized hyperparameters, we get the better quality of predictions. This is followed by RASAR model development. As I mentioned, that RASAR is a combination of read-across and q-SAR, where we use the q-SAR descriptors or structural physicochemical descriptors along with similarity measures. So we use both the q-SAR and RASAR descriptors in the same model. So we get here 4 individual models and some pooled model, and also we get for consensus. Consensus means the combination of model predictions are used to define prediction for query compound. So we get 3 kind of consensus prediction in this one. Now this slide shows a bubble plot for different descriptors. So here the bubble size represents that is actually the reflective of the importance of individual descriptors, and we will see that the RASAR descriptors, which are having the more bubble size, so RASAR descriptors are more important than q-SAR in most of the cases. This graph shows that the highest external validation in terms of q square f1 is obtained from pooled descriptor RASAR models. So pooled descriptor RASAR model shows q square f1 value of 0.671, superseding both q-SAR and read-across. And in case of, if we take the MA value, that is the mean absolute error, which should be minimum, that is obtained from the consensus model in RASAR model, and this is superseding the quality of model of q-SAR and read-across. Now, one more interesting thing, I showed one measure, that is G, or concordance measure. I told that it has some problem. So in the first plot where G has been used, and the VIP plot shows that the G is not much important. But when G is replaced by GM, the modified G, the expression is shown here, it shows the second highest importance. So GM is more important than G. So we use GM in our work, modified G instead of G. In the next work, we have used RASA for machine learning based q-SAR prediction of different toxicity endpoints. Here we have used rat androgen receptor binding affinity, acute oral toxicity data in avian species, specific avian species, acute contact toxicity in honeybee, acute oral toxicity in Melartic and avian species, and inhalation toxicity data. And this also shows the bubble plot showing that RASAR descriptors are more important in most of the cases. Now, this is a comparison of model quality for RASAR and q-SAR. The first row shows RASAR, the second row shows q-SAR, and this is replicated in different datasets. As you can see, the q square f1 is the external validation measure. For dataset 1, q square f1 is 0.70 versus 0.58 for q-SAR model. So RASAR model is superseding the quality of external validation for dataset 1. This is shown in all the dataset. In case of all the dataset, the q-SAR model shows the better quality of external validation than q-SAR models. And this different scenario that we can obtain our query compound may be close to the positive compound, and
Ma'am, your voice is not clear.
So close.
Sir, your voice is not clear.
Maximum positive negative.
Actually, Professor Roy has some connection problem. He will be joining soon.
I have joined soon. Join again.
Okay, thank you, sir.
It was disconnected just.
Sir, again your voice is breaking.
Now. It was decided just now.
So, sir, your voice is not clear. Breaking. Can you please disconnect and rejoin?
Yes, sir. Yes, sir. Voice is not clear.
Hear me now?
Yes, sir. Yes, sir.
Yes. Now.
Okay, okay,
Audible.
okay. Okay, so this is RASAR modeling for hepatotoxicity data set. And after collection of the hepatotoxicity data, we do appropriate structural representation, descriptor computation, and data pretreatment, then data set division. So the training set is used for model development. Test set is kept aside if this is not used for model development. And feature selection is done solely from the training set. After selecting the features, we do the appropriate similarity descriptor computation. And from these cluster descriptors, we again select the important features and we go for machine learning model development. LDA, that is linear discriminant analysis, logistic regression, random forest, and support vector machine. So this we do for both q-RASAR and RASAR. And then we compare the quality of the test set prediction for q-RASAR and RASAR. As you can see that if we consider Matthews correlation coefficient, that shows that the q-RASAR model, the logistic regression shows the MCC, 0.22, that is the highest value. Whereas in case of RASAR, for the test set prediction, logistic regression gives 0.27, whereas LDA gives 0.29. This is the highest value. So the consideration is used for case identification. Now, for doing this, for the set of compounds, we can divide them in two parts, positive and negative, based on the training set pin. And we can normalize the selected descriptors, and we can calculate the average values of those normalized descriptors for both the classes, positive and negative. And then we can find whether the descriptor values are showing high, that is the higher value for the positive class or negative class. So some of the descriptors will show higher value for the positive class, some of the descriptors will show higher value for the negative class. And based on that, we can classify descriptors into groups. And we can calculate weightage based on the formula that is shown at the bottom of the slide. That is the difference of average values for the both classes and divided by sum of different values of all descriptors in a particular class. And then we multiply these weighted values for this with the standardized descriptors, and we can calculate ARKA descriptors, arithmetic residual in K group analysis, in 2 dimensions, ARKA 1 and ARKA 2. Now, when we plot ARKA 2 versus ARKA 1, the positive component should reside in the 4th quadrant and negative component should reside in the 2nd quadrant. But when the opposite happens, that is, if negative component in the 4th quadrant and positive components are in the 2nd quadrant, where those compounds are opposite class, but a particular compound shows activity in the unexpected class. So when we find with small changes in the activity, in the structural features, the activity value differs too much, then we can find out activity claims. So this can be easily identified using this approach by calculating ARKA descriptors. We can give some examples like, say, bupivacaine, which is an hepatotoxic drug, whereas the mepivacaine, which is a close congener, is non-hepatotoxic. This is an example of activity claim. Similarly, dianosterol is non-hepatotoxic, whereas phenol is hepatotoxic. This is another example of activity claim based on their close similarity value. Again, amphotericin B is hepatotoxic, whereas its close source congener nystatin is non-hepatotoxic. This is another example of activity claim. Esomeprazole is non-hepatotoxic, whereas its most close 5 congeners are hepatotoxic. This is another example of activity claim. So this can be easily identified by the ARKA approach, and we have different tools we have developed for our RASAR model development and read across. Quantitative read across, RASAR descriptor calculator, then auto array optimizer for optimization of different hyperparameters, and for different machine learning algorithms. We have different tools which can be downloaded from our site. For the QR code of our site is given in the left top corner of this slide. We have also applied RASAR for different other endpoint values in material science and toxicology, etc. So this slide shows application of machine learning based q-RASAR approach for efficient prediction of specific surface area of perovskites, then efficient prediction of cytotoxicity of titanium dioxide based multi-component nanoparticles using machine learning based q-RASAR approach. So these studies report the development of simple, interpretable, and reproducible q-RASAR models for various toxicity and also activity and property endpoints. The q-RASAR models reported here thus deliver lower prediction error for the query sets than the corresponding q-RASAR models, suggesting that they are potential models of choice for efficient prediction of a given level of chemical information. So based on the variable important plot, in most of the cases we have seen that RA function, GM, and average similarity are the most important similarity-based determination for prediction of toxicity, which warrants further extensive studies on this function. The ARKA descriptors may help to identify activity cliffs, and the combination of ARKA with RASAR can give a good modeling strategy, based on which we have recently modified the RASAR workflow, and the 2nd generation RASAR we use the ARKA descriptors for calculating the similarity basis. As RASAR is essentially a similarity-based approach, identifying activity cliffs may help the RASAR models effectively. The use of RASAR and ARKA approaches in combination may prove to be an effective strategy for predicting model. So the read across and RASAR modeling research were funded by science and engineering research. So acknowledge the funding received from different funding agencies over the years. And this is the book on q-RASAR, published as Stranger Brief. There are several other books on q-RASAR and their application. With the q-RASAR with the application in different fields. This Stranger Brief is a reference book